Within the peripheral nervous system, there are satellite cells and there are Schwann cells. Now, satellite cells will surround the somas within the peripheral nervous system, and this helps to regulate the chemical environment of the neuron, so regulation of gases, nutrients, and neurotransmitters around these particular regions. The Schwann cells will surround nerve fibers in the peripheral nervous system, and they'll form a myelin sheath around the thicker nerve fibers. And these are functionally similar to that of oligodendrocytes. And Schwann cells are integral to the regeneration of damaged peripheral nerve fibers. So it is achievable for there to be repair post-injury within the peripheral nervous system as compared to the central nervous system. And that's the difference with respect to that scar tissue that we see generated in sites of injury within the central nervous system. But for the peripheral, there is actual repair processes that can occur. And when it comes to repair mechanisms from central versus peripheral, this used to be something that was really successfully pursued within a lab at Hunter College. And this was the work of Dr. Mari Philbin. And she was really trying to figure out the process of regeneration in order to help those individuals that suffered damage to their central nervous system. But unfortunately, she passed away from cancer several years ago. So the work of her lab eventually stopped. When it comes to the nervous system processing information, it takes place within 3 stages or 3 steps. There is the sensory input shown in the blue arrow, the concept of integration shown in green, and then motor output shown in red. Now, in general, the nervous system uses sensory receptors to monitor changes occurring both inside and outside of the body, and that information is kind of amalgamated, grouped together, and that's the sensory input. The neurons that are associated with sensory input are afferent, A as in apple, afferent, and will conduct signals towards the central nervous system. Now, during integration, the nervous system processes and interprets that sensory input and then decides what should be done within that moment. Now, these neurons lie entirely within the central nervous system, and this will serve as that interconnection between that of the sensory input and that of the motor output. So that's the outgoing pathway shown in red. Now, the nervous system activates effector organs or motor neurons, and these neurons are efferent with an E as in Edward, meaning that they'll conduct the signal away from the central nervous system. So these will be things such as muscles or glands that will cause a response that's described as the motor output. So for the tropical snail example, there will be sensory neurons that are found on the siphon, which can be used to gather information about the surrounding environment. Is there something in the close vicinity that can be ingested essentially? And then there's the integration step, right? So we're thinking about, okay, here's the information that I've received from the siphon. What will I do with it? How do I interpret it? How does the nervous system respond to that? And in this scenario, the response will be to trigger the release of this proboscis. And the proboscis is a type of harpoon, which will go ahead and anchor itself to the prey and also release those neurotoxins that result in paralysis and ultimately death. So the purpose behind this particular image is just to display the structural diversity of neurons. All neurons will have a cell body, so that's the soma, and one or more processes. So we can see that there are dendrites shown in black, and then axons that are shown in red. Now, in looking at this particular image, this isn't to say that all sensory neurons and interneurons and motor neurons have the specific level of organization, but what you should be able to do when looking at this image is think about how form and function are supported here. So in thinking about an interneuron that has to integrate and relay and convey a bunch of information that it's received from a sensory neuron, there is a ton of extensive branching that's taking place with respect to axons and those resulting synaptic terminals. Whereas the motor neuron, if we think about the relationship between the interneuron and the motor neuron, has a really extensive amount of dendrites, which would allow it to receive a bunch of information from these synaptic ends that are seen in the interneuron. So just think about the interplay between these different neuron types. Again, this is not like a definitive image of how each of these neuron types would look, but it gives you a sense of how, in general, they would be organized and how that organization helps to support their role within the process of information processing. Within Module 2 of Chapter 48, the focus really lies on resting potential. And so we'll go through resting potential and the components that help to establish that state within the second module. And then you're going to want to connect what's taking place with respect to resting membrane potential with the action potentials and graded potentials that we'll discuss in the next module. Prior to thinking about the specific events at the neuron, meaning what is being mentioned within concept 48.2 and the rest of the slides within this module, I want to mention that our body overall is electrically neutral. There are equal number of positive and negative charges. But there are areas in our body where one charge is seen in greater amounts than another, meaning there are areas that are positively charged or negatively charged. We have discussed throughout this 2-semester course the idea of opposite charges having a level of attraction towards one another. And if opposite charges are attracted to one another, that means that work must be done in order to keep those charges apart, if that's the goal. So this means that any situation in which there is intentional charge separation is an indication of potential energy, energy that we can harness to do something with. And this is essentially what's seen at the plasma membrane of the neuron. So while we're focusing on the neuron, this is not the only area within our bodies in which we can utilize this charge separation, but I do want you to just have an overall understanding of the fact that our body is not going to be doing the same thing in each place, and also that how things are distributed within our body is also going to deviate, and that deviation is really going to help to facilitate whatever role or events are meant to be taking place within particular systems or cells or tissues or whatever the case may be. Concept 48.2, ion pumps and ion channels, establish the resting potential of a neuron. So every cell has a voltage, and a voltage is established due to a difference in electrical charge, so meaning a difference in the ions that are seen across the plasma membrane, so meaning outside of the cell versus inside of the cell. And that's referred to as membrane potential. So again, it's really important to keep in mind that membrane potential is based on the specific voltage differential seen directly across that bilayer and not just the overall amount of ions outside the cell and inside the cell. We're looking at a discrete region. And the kind of nature of this lends the neuron to a level of excitability when there are deviations or fluctuations seen across that plasma membrane. Now, while I'm mentioning the neuron right now, every cell has that voltage. So this is a concept that was raised within Chapter 7. So it's same conceptually. We can establish a voltage or charge due to a difference of ions across the phospholipid bilayer. Now, when we're thinking about neurons, then we can use terms such as resting potential and action potentials. Now, a resting potential would be a neuron at rest. And in the sense of neurons resting, what we're saying essentially is that they're not sending signals, because recall that neurons are really focused on communication, and communication occurs through the receipt and transduction and continued transfer of those signals. So a neuron at rest is not going to go through that specific process. But once the right situation occurs, when the conditions are appropriate, then an action potential can be elicited. And the way that we can elicit an action potential is through changing the membrane potential. So there is a level of sensitivity to the voltage found at the membrane, such that we can manipulate the ability for a neuron to elicit a signal based on changes that occur across that bilayer. So that's the property that we'll think about in this module, but then we'll continue to apply in the next, because again, we are breaking up resting potential and action potentials as far as your textbook goes. Table 48.1 provides us with information about ion concentrations inside and outside of a mammalian neuron. Now, while it provides us with physical numbers, these numbers may be slightly different from other books that you look at that are based on the same topic and also introductory in nature. But what's important to take from this is that the ions that are critical for establishing the resting membrane potential are going to be potassium ions, sodium ions, and chloride ions. Now, I won't say much about chloride ions apart from acknowledging that they are involved in establishing resting membrane potential, but they do so to a lesser extent. Now, in looking at this particular table, there's also the presence of large anions that's kind of referenced here. And I will discuss this a little bit more in a different voice thread comment, but this will also play a role in the resting membrane potential just based on electrostatic attraction. So some properties to consider when it comes to thinking about the role of resting membrane potential is that of electrostatic attraction, the idea of diffusion, and how these events are going to influence what the charge will be across that phospholipid bilayer. So here we're focusing on our nerve cell, and nerve cells have voltages. And voltages are something that we can have as a quantifiable property, and it will be a measurement of potential energy based on separation of charges, which is a concept that we talked about during Bio 100 in Chapter 7. So the separation of charges across the plasma membrane is called the membrane potential. And we're focusing on the nerve cells, but every cell has a voltage. Now, resting potential is the membrane potential of a neuron that is not sending signals. And it develops because the cytosol of the cell compared to the extracellular fluid have different compositions of ions. So there's an imbalance of sodium ions. So we see based on the key that there's far more sodium ions in the extracellular fluid than the intracellular fluid. And for potassium ions, we see there's far more within the intracellular fluid than the extracellular fluid. So there's an imbalance of these ions as evidenced by the picture. And what's not shown in the picture are trapped anions. So things that will have a negative charge, like macromolecules such as proteins and RNA and other molecules. that will be found on the cytosol side, so the intracellular fluid side, but will either be significantly reduced or completely absent from the extracellular side, so outside of the cell. Now, these molecules, and this is something that we also learned in Bio 100, can't pass through the plasma membrane, but they can exert a force on ion movement because their charge can exert a level of attraction and kind of help to hold on to those opposite charges, keeping them from diffusing down their gradient. So resting membrane potential is really due to unequal distribution of components found on either side of the phospholipid bilayer, and factors that help to influence resting membrane potential are the concepts of diffusion, so diffusion down a concentration gradient through the membrane. And here we see that there are channels with specificity for these ions. We said that there was a lot of potassium inside, so it will have a tendency to move down its gradient into the extracellular fluid. Sodium ions will have a tendency to move down their gradient from the extracellular fluid into the intracellular fluid. And even with this movement down its gradient, there is selective permeability of the membrane, which means that some ions pass more easily than others through these particular channels. And then there's also the role of electrical attraction of cations and anions, so opposites attract to one another. And another way of describing this is as electroneutrality. So changes in membrane potential can act as signals, and that can result in transmission and processing of information. So when we're looking at this neuron at rest, the concentration of potassium ions is highest in the intracellular fluid, while the concentration of sodium ions is highest in the extracellular fluid. Now, in addition to these channels that are allowing for the movement of ions down their respective gradients, we also have a sodium-potassium pump, which you learned about in Bio 100 as well, which is also going to be key to its contribution towards resting membrane potential. So we have leak channels, which are these ones that are here in open conformational state. So these are transmembrane proteins that allow for ion conduction through the lipid bilayer. They will be in an open state, which will allow for diffusion into or out, depending on the voltage and depending on the local ion concentrations. And then you will also have your sodium-potassium pump. And as a reminder, but we'll go through the process again, the sodium-potassium pump is going to require hydrolysis of ATP, and it's going to be driving movement of ions against their concentration gradient. So we'll be moving 3 sodium ions out into the extracellular fluid, 2 potassium ions into the intracellular fluid. So this is happening at the neuron, and this contributes to the resting membrane potential, the value of the neuron across this bilayer specifically while it is at rest, while it is not conducting any information. What we have on this particular slide is an image that is pretty much identical to one that is covered in the first half of this 2-semester course, and that is the sodium-potassium pump. Now, something that I often say when it comes to different topics that we discuss along the way is that we can talk about the same thing multiple times throughout the duration of a semester or duration of a year, and what changes is our approach, or what changes is now we can add in a layer to the information that we have. Now, what I'm going to do here is really just provide a reminder of how the sodium-potassium pump works, but then just keep in mind that the sodium-potassium pump's role in this particular scenario is to help contribute to that resting membrane potential value. Now, the leak channels are going to play a really significant role in that, but the sodium-potassium pumps are going to be the component that's going to utilize energy in order to contribute towards the resting membrane potential. Now, let's just work our way through, starting from 1 and ending at 6. So when we take a look at step 1, we can just acknowledge what's going on with respect to ions on either side of our phospholipid bilayer. So we have a low concentration of sodium ions in the cytoplasm, so on the intracellular side, and a high amount of sodium ions in the extracellular fluid. This is going to be an example of active transport because we are going across our concentration gradient from a region of low sodium ions to that of high. When we take a look at our protein, and this is a transmembrane protein which spans the entirety of the phospholipid bilayer with a portion that extends into the cytoplasm and a portion that extends into the extracellular fluid, we find that there are 3 binding sites for our sodium ions. So in panel 1, we have 2 sodium ions that are already bound. We have a third that is about to come in, and that's going to be the final sodium ion in terms of being completely occupied and allowing us to transition from step 1 to step 2. So we have our sodium-potassium pump still facing the cytoplasm, now containing 3 sodium ions in each of those sites. So there is a high affinity or attraction of those sodium ions to these 3 binding sites within the protein. Now, once we have our fully occupied state, we can see that we're going to have ATP hydrolysis. ATP hydrolysis is going to result in the formation of an adenosine diphosphate and this phosphate group. And this phosphate group will be conjugated or bound to the sodium-potassium pump while it's in this current conformational state. So now we're going to think about the impact that that phosphorylation event has on the protein. The impact of that phosphorylation event is to induce a conformational shape change in our sodium-potassium pump. So notice that as we transition from 2 to 3, that we go from having a protein facing the cytoplasm to one facing the extracellular fluid. Now, mechanisms that are beyond the scope of this course cause that conformational shape change, and there's also a shift in affinity or attraction of the sodium ions for these binding sites, and so they will dissociate, and they'll dissociate and enter the extracellular fluid. So now we've allowed for 3 sodium ions to go against their concentration gradient and enter the extracellular fluid. While our protein is facing the extracellular fluid, there will be potassium ions, and those potassium ions will need to be transported against their concentration gradient into the cell. So as a reminder, our potassium ion concentration outside of the cell is low, but inside of the cell is high, which means we're depending on the utilization of energy in the form of ATP to establish movement, and that's applicable to both the sodium ions and the potassium ions. Now, while we're facing the extracellular fluid, there is a high affinity or attraction of these potassium ions for the binding sites that are here, and there are just 2 compared to the 3 that are seen for those sodium ions. So we'll have our potassium ions coming in, binding to the 2 respective sites, and then we're waiting for them to become fully occupied before we transition to the next stage. But something to notice that when we are at step 4 is that there needs to be some type of hydrolysis event in order to remove that phosphate group or that phosphorylation event that allowed for the conformational shape change of that protein to occur. And that conformational shape change was going from the cytoplasm facing to the extracellular facing state. So once we had a fully occupied state, so once 2 potassium ions had come in and bound to those 2 respective sites with a high level of attraction or affinity, we had the hydrolysis of our phosphate group, and that will remove the phosphate from the protein. Now, in the absence of that phosphorylation, the protein is going to convert back to its original state, which was cytoplasm facing. And in the cytoplasm facing state, we are going to see a decrease in the affinity of those ions for the binding sites, and that's what's going to allow the potassium ions to enter the cell. So through this process, we're able to move 3 sodium ions out of the cell and 2 potassium ions in, so we have an unequal distribution of ions across our plasma membrane. So Nernst equation or Nernst potential describes the relationship between membrane potential and ion concentration, and we generate a value for Nernst potential for each specific ion. So in looking at the equation that's down here, the math doesn't matter as much as the results that we get through this equation, which is that the Nernst potential for potassium is very negative, while that of sodium is positive. Because the density of potassium leak channels found along the neuron compared to sodium is different, there's far more potassium leak channels than there are sodium, the Nernst potential for potassium has a higher contribution towards what is considered the resting potential of a neuron, which, depending on the neuron in question, can be something like -80 or -70. It really does depend, but it will never be exactly -90 because we have to account for the behavior of sodium ions. So potassium exerts the greatest influence between the membrane, and it is more permeable to it. So potassium is more permeable to the membrane than sodium ions. So that was one of the factors that contributed to the resting membrane potential. So if you have potassium ions diffusing down its concentration gradient and heading out of the cell, it's going to leave cytoplasmic anions behind. And recall that those are things that are incapable of making their way out of the cell, so they're stuck on the inside. And this will exert an attractive force on these ions. And essentially, once a balance is reached between the desire to diffuse down a gradient versus the electrical attraction for what remains inside, we find that it leaves many more times of concentration for potassium in the intracellular fluid as compared to the extracellular fluid. And when it comes to this equation, the numerator reflects the concentration of the ion seen on the outside. The denominator reflects the concentration of that particular ion seen on the inside. For the sodium ions are far more concentrated on the extracellular side, so within the extracellular fluid, which would be the numerator here. It's still capable of diffusing down its gradient, but it's less permeable, and there's far fewer of these leak channels present, so there's a small amount of trickling out that occurs. And when it comes to this setup, you can see that you have a chamber, inner chamber, outer chamber. So this is not really meant to reflect true conditions, right? We're doing something in vitro here to kind of get an understanding of how these ions behave. But something that's important to keep in mind is that because we see these differences in terms of the behavior of each of these particular ions, anything that's going to kind of modify or fluctuate the movement of these ions is going to have an impact on the voltage that's seen across that membrane. Concept 48.3, action potential.